Humans differ from the rest of animals in many ways, but what really sets us apart, even from the other great apes, is our brain. Our brains are larger and much more complex, so they can support more sophisticated functions such as abstract thinking, the planning of actions, very special memory and learning abilities, a complex language, cooperation, all of which were the basis for the creation of tools, technology, and culture. All these human-specific characteristics have evolved in the last 6-7 million years since the split from our common ancestor with the chimpanzees, who are our closest relatives in the tree of life. As usual, fossils tell us something about the difference between humans and the other great apes, and in some cases they can tell us when such differences appeared within the human lineage. Comparing fossils of the skulls has shown, for instance, that brain size increased extraordinarily throughout this period, but it is difficult to find in fossils details about the changes in the internal structure and functioning of the brain or changes in embryonic development that lead to such complex brains. Again, the comparison of genomes can tell us a lot of things about this process that we could not know otherwise. When the complete sequence of the genome of humans and chimps was deciphered, scientists thought that it would be relatively simple to find those changes that explain what makes us human, those genomic innovations that are unique to us. However, the degree of similarity between our genome and the chimpanzee genome is surprisingly high, at least considering the striking differences in morphology and behavior. It is usually emphasized, for instance, that around 98.5% of the letters in the same position in both genomes are identical. And almost 1/3 of proteins have exactly the same amino acid sequence in humans and chimps. But this still leaves a lot. This leaves about 20 million genetic changes that are specific to humans. The thing is that most of them have no functional impact, at least that we can recognize today. So the question is, from those that might have functional implications, how do we identify the ones that are more relevant, and how do we link them to specific phenotypic changes? Well, research over the last years has found some interesting candidates. For instance, scientists have found some primate-specific genes that originated during the evolution of hominoids. A couple of genes in this category were created by retrotransposition, a mechanism that we already covered in a previous unit. One of them is GluD2, a copy of a gene called GluD1. GluD2 is active in mitochondria and helps to recycle glutamate. Glutamate is an important neurotransmitter, so this could have led to changes in brain function in primates. Another example is the expansion of a gene family of transcription factors with zinc fingers, for instance, the gene ZNF674, a member of this family. This gene is also specific for primates and is implicated in mental retardation syndromes in humans, so it could be important in cognition. But then, of course, what we really want to know is which changes are specific to humans as compared to great apes. We know of some genes that have lost their function in our lineage. They work in chimps, but during human evolution, these genes have been inactivated by various mutations. One of them, called CMAH, encodes an enzyme which is necessary to make a type of neuraminic acid, a sialic acid present on the surface of the cells of other primates and mammals, which we seem to have lost, so our cells don't have this sialic acid on their surface. Or the gene for a type of myosin, the protein that forms muscle. It seems that the loss of the gene coding for a particular type of myosin, myosin 16, in humans led to our smaller jaw muscles, and that would have made possible to have a larger cranium and a larger brain. But how about true genetic innovations? Genetic innovations that are unique to humans. Let me tell you briefly about two examples that have drawn a lot of attention in recent years. In 2001, a gene called FOXP2 was found to underlie a rare inherited speech and language disorder. The gene encodes a transcription factor, a protein that binds to various sites in the genome in order to turn on and off a variety of genes. The protein FOXP2 is highly conserved among mammals, but in humans it carries 2 amino acid changes at positions 303 and 325. And these substitutions occurred after the divergence of humans from their common ancestor with chimpanzees, and we know this because these 2 changes are not found in the chimp genome. Obviously, people thought that the human variant of this gene might have a lot to do with our ability to use complex language. So scientists did very nice experiments. For example, they inserted the human version of the gene in mice. And what happened was that the mice changed their brain somehow. Human FOXP2 protein improved learning in mice and promoted the formation of synapses, the connection between neurons. As I said, FOXP2 is a transcription factor, so it makes all these changes by regulating the activity of other genes. And what is amazing is that when scientists knocked out one of these genes in mice, they observed a decrease in the number of ultrasonic calls of pups to their mothers. All this supports the notion that the peculiar human version of this gene and its protein might explain why humans are capable of language and chimps are not. Then, in recent years, scientists could read the genome of 2 closely related ancient humans, the Neanderthals, who lived here in Europe, and the Denisovans, who lived in Siberia. We will talk about Denisovans a bit more later on. But what matters now for the FOXP2 story is that it was generally assumed that Neanderthals did not use language, at least a language as complex as ours. So it was a great surprise when scientists looked at the sequence of the FOXP2 gene in these ancient genomes and found that both Neanderthals and Denisovans had the same 2 mutations, the same 2 amino acid changes as modern humans. So they probably were capable of some basic form of language. In fact, they had some degree of cultural development, social organization, tools. So it is quite reasonable that they had some form of language, because language is required for all those activities. So we have some genes like FOXP2 which have a few mutations exclusive to humans. But what about entire new genes? Are there any genes out there which really set us apart from the rest of great apes? Well, I'll finish this presentation telling you the story of the only example we have so far of an entire human gene that is not found in the genome of the chimpanzees or in any other genome for that matter. This gene is called MER941 because it codes for the precursor of a microRNA. And microRNAs, as you probably know, are small RNAs that regulate negatively the expression of protein-coding genes. They usually switch off specific genes. It seems that MER941 has evolved after the human and the chimpanzee lineages diverged about 6-7 million years ago. The genomes of present-day humans have between 2 and 11 copies of this gene, with an average of 6 to 8 copies. Interestingly, the genome of Denisovans, those archaic humans found in Siberia, already contained at least 2 copies of this gene. So what is the function of this microRNA? What genes are regulated by MER941? It seems that in humans it regulates genes involved in 2 main signaling pathways related to neurotransmission, which again supports that this gene has something to do with neuronal function. In fact, these human-specific effects are detectable in the brain, and a small deletion of the region that includes MER941 results in mental retardation and speech and language defects in humans. So this tiny, unique human gene might also have contributed to these human-specific features.